So one of the fundamental goals of the study was actually to understand differences of sarcopenia across ethnicity. So we really wanted to cover different people across the globe. So we sampled people in Singapore, in Jamaica, and in England. And the fundamental difference we observed is really the bioenergetic function. So when we asked openly what out of our 25,000 genes are differently expressed in sarcopenia, the vast majority was everything linked to mitochondria. We believe it's an intrinsic change that happens within the cells. So the ability of mitochondria to turn our nutrients, so the nutrients reach the cell, they get digested in our gut, they reach the cells, and after that they need to get converted in energy under the form of ATP. And this happens through the metabolic reactions in the mitochondria and ultimately through the electron transport chain. And what we observed in sarcopenia is really all the complexes of this electron transport chain were inefficient, were low in activity, and therefore they couldn't turn the same amount of energy, of nutrients into energy, Well, I think that there's really two big questions in that concept. The one thing we need to understand is the amount of energy that can be produced at a given input of calories, basically. And this is in part due to very active, fast metabolism of using sugars and glucose that can be independent of mitochondria. But there's also the more long-term production of energy that's really driven through the mitochondrial process of generating ATP for cellular reaction. So we think that's the first question, is what's the amount of energy and the efficiency of metabolic coupling that needs to be addressed. And of course the second one is how do we use the substrates differently, because the food we eat is not homogeneous, every calorie is not the same, and so using proteins, sugars, or lipids is not going to generate the same sort of cellular adaptations. So we think that's the second part of the question, and we're looking into ways to recouple both of these processes with nutritional interventions. NAD is a substrate that's within our cells, and it's basically a shuttling molecule. All it does is it uses enzymatic reactions to load protons, and this generates energy. It's a way to carry energy, if you want, in the cell. And so the mitochondria needs nutrients to turn NAD plus into NADH, and this reaction will then allow the mitochondria to use NADH to generate energy. So it's a shuttle, if you want. But it's also used by certain enzymes, so the absolute levels of this pool can vary, and we know with aging there's variations and there's decline in this total level of NAD. And of course, if the shuttle declines, then the ability to shuttle this energy doesn't work as well as when we were young. And specifically, that's what we observed in sarcopenia. The muscle of sarcopenic patients had a lower capacity to replenish this total pool of NAD, and when we measured this enzymatically, we saw lower levels of NAD associating with sarcopenia and with muscle strength and muscle function. The big trend is really to see a decline in NAD levels with age, but we have to be cautious with the interpretation, because of course NAD is a fundamental constituent of every cell in our body, but the regulation is very tissue specific. So what happens in different tissues can vary. A lot of what we know is actually either from model organisms, where there it's very clear that NAD levels decline in most tissue. The rate of decline is actually a bit dependent on which tissue you look at. The high metabolic rate tissues tend to be more affected. But also the tissue specificity makes it difficult to study in humans. And in our case, studying muscle, we're quite in a favorable situation, because we can collect biopsies and interrogate directly the target tissue in humans. But for people looking at brain, it's impossible, of course. And other tissues, you can do it to some extent, but it's much more complicated. So we were one of the first to actually prove this directly in a tissue in humans. Other work has shown either in skin biopsies or in blood cells that the concept of the decline is also possible in humans. But again, we have to realize this happens when you compare many patients. There's also inter-individual variability and inter-tissue variability. So it's really a trend that we observe when we regress analysis over many patients. So I think there's different layers of impact, and it's hard to make a very causal assumption in a very direct way, but the fundamental thing we know is NAD is required for all these energetic enzymatic reactions. So lowering NAD level is believed to associate with lower bioenergetic and lower energy production within our cells. And then indirectly it affects the function of our mitochondria, and we know different tissues can respond a bit differently to mitochondrial efficacy, and in particular it's really the tissues that have high metabolic rate that will have a high need on mitochondria. So in muscle, which we study, of course, the key function here is contraction, and we need a lot of energy because to turn the calories into power, into really contraction. Other tissues, for example, in kidney, you need a lot of pumping of the metabolites and the electrolytes that are filtered by our kidney cells, and this is very high on mitochondria, so their NAD plays also an important role, and so on. The more the tissue is metabolically active, the more the NAD will have an impact. I mean, one of the findings in preclinical models is really that it can reverse several of the manifestations of aging. So that's the interesting thing about the approach, is because it's a conserved molecule and this decline happens, although at different levels, it happens in different tissues. The applications are widespread across different tissues where the mitochondrial turnover is high. So obviously, based on our data, we think helping NAD and mitochondria in muscle will be important to maintain muscle function as people age. Of course, at this stage, it's largely association from observation. So what needs to be done and in humans in the clinic is really to go for interventions where we restore these levels or prevent these levels from declining and show that this has benefits. And this work has been done in model organisms, and the results are promising, but now is the exciting time where these interventions are transitioning into clinical testing. Well, NAD is very sensitive to dietary intake, and you can influence the levels at different levels. And the first one is tryptophan, which is an amino acid from the protein we eat, is an indirect precursor. Vitamin B3 that we also find in our diet also feeds into the NAD pathway. So in a sense, NR is not fulfilling a completely unique role. Where it's very powerful is actually that it could be converted to NAD in a very direct transition. So it's much closer in the enzymatic reaction to NAD than all the other precursors that are more indirect. And the reason why we think it's important for sarcopenia is specifically because the enzymes that are required for tryptophan or vitamin B3 to be converted to NAD are actually altered in sarcopenia, whereas the enzyme that converts NR, nicotinamide riboside, to NAD is intact. So we believe in this case it's a better way to reverse the deficiency that we're observing. Well, I think again it's back to the question of how we couple calories. So the protein especially in muscle, has two roles. First, it provides energy, and like any other cell of our body, we can use the amino acids as a source of energy. But in muscle, there's a second and very specialized role, and that's why most sports people, athletes, as well as older people consume protein, is because protein is the main constituent of our muscle. The way muscle contracts is by accumulating what we call contractile protein that can contract when our brain instructs them to do so. And we know the muscle mass is basically governed by the amount of these contractile protein, which is influenced by our dietary protein intake. And this whole regulation is very plastic and dynamic. So anabolic and catabolic tissues will regulate how much the dietary protein translates into muscle protein. And of course, there's hormonal cues, and the system is much more complex. But that's the fundamental reason why we give protein is to maintain this muscle mass and to provide the building blocks and to stimulate the anabolism, because there's sensor systems in our body that sense how much amino acids we have and that regulate protein synthesis in response. So that's the key role of proteins. Now, where we think cellular nutrition can bring an added benefit is really because with age, there's a lot of stress that accumulates in our cells. There's a lot of reactions that basically decline in intensity. So by fixing these, and we can do this at different levels, it can be oxidative stress, it can be NAD biology, it can be even just the way to better recycle the materials. We know like autophagy, which is cellular recycling, actually declines with age. So the intervention on these cellular nutrients basically allows to fine-tune how our cells turn these macronutrients, these calories, these proteins, into beneficial effects. And so we think these benefits will not be direct, basically. They'll just allow to better prime the machinery and make it youthful again, in a sense. Well, you know, as a muscle biologist, I have to answer mobility and muscle performance, because this is one of the main physiological declines that happens with age. We know peak muscle mass and performance happens in our 30s, and we all notice a slight decrease, but really this becomes a major problem in late life when people that don't train enough and that are too sedentary start losing independence. So this is definitely one, but of course I can't ignore the rest of the literature from all my colleagues working in different organs. And there's many organs where mitochondria play a fundamental role. In the heart, we know this is a highly rich organ in mitochondria, and the heartbeats and the contractions need efficient coupling. As I mentioned kidney previously, we know there's good evidence there that the mitochondrial decline is important for the urinary filtration. The brain is probably another one. We know there's actually genetic associations that link deficits in mitochondrial processes, and in particularly into the ability to recycle mitochondria to neurodegenerative disease, and especially Parkinson's disease. So in a nutshell, we think the benefits are wide. They're largely focused on tissues with high metabolic rate that use a lot of mitochondria to convert the fuels into energy. Well, like in many biomedical conditions, biomarkers, I think, are really the future to understand how a different person will react to a different intervention or therapy. So there is a future to biomarkers, I think. With mitochondria, I think one of the challenges is still that these are intracellular organelles, and so you cannot easily read out mitochondrial activity without having access to a cell in our body. So that's really the limitation today. You can do it to some extent in blood, but the technologies to measure mitochondrial function and mitochondrial activity are not widely available for clinical or medical practice. So today we're really at the level of research. There's a few candidates for biomarkers, so there's surrogates, there's molecules that mitochondria will make and spit out in the blood that can be detected, but it's quite an early field, so we're not at the stage of clinical implementation. As researchers, we follow this, we try to implement these measures in our clinical studies and in our preclinical studies, and we really hope in the future this will allow us to stratify the populations, because ultimately, well, first, mitochondria is a very wide cellular space, and there's many different ways this mitochondrial processes can go wrong. So having specific biomarkers would be very important. The specificity and their sensitivity, as well as the ability to measure it in available matrices, will be really what drives the implementation in the long term. Well, no, I think we're quite excited to see our research that was exploratory translate and into areas of biology where there's nutritional interventions. So we're looking forward to seeing these interventions transition to being available to people and transition to clinical demonstrations.